What makes us who we are? For centuries, this question has captivated minds. Today, we can unlock secrets within our cells. This is the era of the genetic revolution, manipulating the building blocks of life, our DNA, exploring history, science, and ethical dilemmas. Genetic manipulation captivates scientists and writers. Fantasy is rapidly becoming reality, treating diseases and enhancing capabilities. With power comes profound responsibility. It's about what it means to be human, choices, boundaries, and legacy for generations. Join us in exploring the genetic revolution. Imagine a vast library filled with countless volumes, each page containing the instructions for building and maintaining a human being. This is our DNA, the blueprint of life itself. Within its intricate structure lies the code that determines our traits, our predispositions, and even our vulnerabilities. Understanding this code is key to unlocking the potential of genetic manipulation. Our DNA is made up of genes, segments of code that act as instructions for building specific proteins. These proteins are the workhorses of our cells, carrying out a myriad of functions that keep us alive and functioning. By altering these genes, scientists believe we can treat diseases, enhance desirable traits, and even potentially extend human lifespan. But how did we come to unlock this knowledge? How did we go from observing the inheritance of traits to manipulating the very code that governs them? The story of genetic manipulation is a testament to human curiosity and ingenuity. From Gregor Mendel's groundbreaking experiments with pea plants to the discovery of the double helix structure of DNA, scientists have painstakingly pieced together the puzzle of heredity. This journey has led us to the development of powerful new tools like CRISPR-Cas9, a revolutionary gene editing technology that has the potential to change the face of medicine and beyond. The ability to edit our own genetic code is a double-edged sword. On one hand, it holds the promise of eradicating devastating genetic diseases, offering hope to millions afflicted with conditions like cystic fibrosis, Huntington's disease, and sickle cell anemia. Imagine a world where these diseases are relegated to the history books, where future generations are spared the suffering and heartbreak they cause. On the other hand, the power to alter our genetic makeup raises profound ethical questions. What are the limits of genetic intervention? Is it ethically permissible to enhance human capabilities, potentially creating a divide between the genetically enhanced and the unenhanced? And what about the unforeseen consequences of tinkering with such a complex and interconnected system as the human genome? These are not questions with easy answers. They require careful consideration, open dialogue, and a deep understanding of the science involved. This documentary will delve into these complex issues, exploring the potential benefits and risks of genetic manipulation with experts from various fields. We will hear from scientists on the cutting edge of this research, ethicists grappling with the moral implications, and individuals whose lives have been touched by the power of genetic technology. Our journey into the heart of genetic manipulation begins not in a state-of-the-art laboratory, but in a humble monastery garden in the 19th century. Here, a monk named Gregor Mendel conducted groundbreaking experiments with pea plants, unknowingly laying the foundation for the field of genetics. Mendel noticed that traits like flower color and seed shape were passed down from one generation to the next in predictable patterns. Mendel's discoveries about inheritance would later become known as the fundamental principles of heredity. He proposed the concept of discrete units of inheritance, which we now call genes. These genes could exist in different forms, known as alleles, and some alleles were dominant over others. Mendel's insights sparked a scientific revolution that continues to this day. The implications of Mendel's work were profound, opening the door to selective breeding. Farmers began to selectively breed plants and animals, crossing individuals with desirable traits. From drought-resistant crops to high-yielding dairy cows, selective breeding has played a pivotal role in feeding a growing global population. The mid-20th century witnessed a scientific breakthrough that forever changed our understanding of life: the discovery of DNA structure. In 1953, Watson and Crick proposed the double helix model using data from Franklin and Wilkins. DNA resembles a twisted ladder with pairs of chemical bases. adenine, thymine, guanine, and cytosine. This discovery revolutionized our understanding of heredity and genetic information. It ignited an explosion of research in molecular biology. The double helix discovery marked a turning point, ushering in an era of genetic exploration. Armed with a deeper understanding of DNA, scientists began developing new tools to manipulate the genetic code. One of the earliest breakthroughs in genetic engineering came in the 1970s with the advent of recombinant DNA technology. This revolutionary technique allowed scientists to cut and paste DNA from different organisms, creating hybrid molecules. Imagine taking a gene from one organism and inserting it into the DNA of another. The implications of recombinant DNA technology were profound. One of the earliest applications was the production of human insulin. Scientists inserted the human insulin gene into bacteria, transforming them into efficient producers. This breakthrough revolutionized diabetes treatment. The ability to manipulate DNA at the molecular level gave rise to genetic engineering. Scientists could now directly alter the genetic makeup of organisms, introducing new traits or correcting defects. In agriculture, this meant developing crops that could withstand harsh conditions and resist diseases. In medicine, genetic engineering held the potential to revolutionize the treatment of genetic diseases. However, this power also raised profound ethical questions about human enhancement and the definition of humanity. The Human Genome Project, mapping our genetic landscape. The completion of the Human Genome Project marked a watershed moment in genetic research. Launched in 1990, it mapped the entire human genome, deciphering 3 billion base pairs. Completed in 2003, it provided a blueprint of our genetic makeup. This revolutionized our understanding of human health and disease. It enabled the development of new diagnostic tools and targeted therapies. The era of personalized medicine began, tailoring treatments to individual genetic profiles. This promised more effective treatments with fewer side effects. However, it also raised ethical questions about privacy and discrimination. Enter CRISPR, a new era of genetic engineering. The story of genetic manipulation takes a dramatic turn with CRISPR-Cas9. This gene editing tool offers unprecedented precision and efficiency. CRISPR-Cas9 is often compared to molecular scissors, capable of making precise cuts in the genome. This precision has revolutionized genetic engineering. Section 2. The mechanics of CRISPR precision gene editing. CRISPR-Cas9's simplicity and versatility are its strengths. It consists of a guide RNA and a Cas9 enzyme. The guide RNA acts like a molecular GPS, directing Cas9 to a specific DNA sequence. Once there, Cas9 makes a precise cut in the DNA strand. This cut triggers the cell's natural repair mechanisms. Scientists can exploit this to introduce specific genetic changes. They can disable a gene or insert a new one. This precision has revolutionized genetic engineering. Scientists can now target genes with unprecedented accuracy. Applications span medicine, agriculture, and the environment. CRISPR-Cas9 is now indispensable in research. It has democratized gene editing, making it accessible to more researchers. This has accelerated discoveries in gene therapy, drug development, and synthetic biology. The technology's ease of use and affordability have led to rapid advancements. Section 3. CRISPR in action, from agriculture to medicine. CRISPR-Cas9 impacts fields from agriculture to medicine. In agriculture, it's used to develop pest-resistant and drought-tolerant crops. Imagine crops thriving in harsh conditions, reducing the need for irrigation and pesticides. Or crops that are more nutritious, combating malnutrition. In medicine, CRISPR-Cas9 holds promise for treating genetic diseases. Gene therapy could correct or replace faulty genes in patients. Clinical trials are underway for disorders like sickle cell anemia and cystic fibrosis. CRISPR-Cas9 is also used to develop new diagnostic tools and therapies for infectious diseases. It can create tests to rapidly identify pathogens like COVID-19. It's being investigated to combat antibiotic resistance. Beyond agriculture and medicine, CRISPR-Cas9 addresses environmental challenges. Scientists are developing biofuels from algae and engineering microbes to clean pollutants. The technology's versatility sparks constant innovation. Researchers are finding new ways to harness CRISPR-Cas9's power. Section 4. The CRISPR Revolution, Transforming Biological Research. CRISPR-Cas9 has revolutionized genetic engineering and biological research. Its speed, precision, and affordability accelerate discoveries in biology. Scientists can easily create genetically modified organisms to study gene functions. This deepens our understanding of complex biological processes. CRISPR-Cas9 enables new disease models for studying health and disease. It revolutionizes drug discovery by identifying potential drug targets. Personalized medicine benefits from studying individual genetic variations. CRISPR-Cas9 democratizes gene editing, making it accessible to more researchers. This has led to an explosion of research and new applications. The technology's transformative impact on biological research is undeniable. It continues to drive innovation and discovery at a rapid pace. Section 5. The Ethical Frontier. CRISPR's Potential and Perils. CRISPR-Cas9's power raises profound ethical concerns. Germline editing could pass genetic changes to future generations. This technology could eradicate inherited diseases but also create designer babies. Unintended consequences are a major concern. Off-target edits could have unforeseen harmful effects. Ethical implications require careful consideration and open dialogue. Balancing innovation and risk is crucial for responsible use. Scientists, ethicists, policymakers, and the public must engage in this dialogue. Responsible use of CRISPR-Cas9 is essential for its benefits to humanity. The ability to edit the human germline using CRISPR-Cas9 has ignited ethical debates. Imagine parents selecting their children's traits like options from a menu. This technology is rapidly approaching reality. Proponents argue it can prevent inherited diseases and alleviate suffering. Germline editing could eradicate diseases like cystic fibrosis and Huntington's. Critics fear it could lead to eugenics and social inequalities. Enhancing physical or cognitive traits crosses an ethical line. This could create a genetic underclass and exacerbate social divides. The debate highlights profound ethical dilemmas in genetic manipulation. Alleviating suffering must be balanced with preventing social injustices. Open dialogue and careful consideration are essential. Ensuring responsible and equitable use of these technologies is crucial. Germline editing affects future generations without their consent. Altering an embryo's genetic makeup impacts all future descendants. This raises questions about intergenerational autonomy. Some argue parents already make impactful decisions for their children. Germline editing could be another tool for providing a better start in life. Critics counter that genetic alterations are permanent and heritable. Unforeseen consequences could cascade down family lines. This could lead to new genetic disorders or alter human evolution. Lack of consent from future generations is a major ethical issue. Critics call for a moratorium on germline editing. Broader societal consensus on its ethical implications is needed. Responsible use of germline editing requires careful consideration. CRISPR technology raises concerns about social inequalities. Access to CRISPR-based therapies may be unevenly distributed. This could create a genetic divide between the wealthy and the poor. The wealthy could enhance their children, widening the gap. A genetic underclass could emerge, exacerbating social divides. Ensuring equitable access to CRISPR is crucial. Affordability, accessibility, and education must be addressed. CRISPR should not be used for eugenic purposes. History shows the dangers of such ideologies. Responsible use of CRISPR technology is essential to prevent discrimination. Section 4. The Unforeseen. Navigating the unintended consequences of gene editing. Predicting long-term consequences of genetic modifications is challenging. The human genome is interconnected, and small changes can have ripple effects. Unintended consequences could arise from genetic edits. Disabling a gene may affect other biological pathways. Germline editing's unintended consequences could impact future generations. Off-target edits are a major concern. CRISPR-Cas9's precision has improved, but off-target edits still occur. These edits could have harmful consequences. Rigorous safety testing and monitoring are essential. A cautious approach to gene editing is necessary. Acknowledging the limits of our understanding is crucial. Section 5. Governing the genetic revolution, balancing innovation and responsibility. CRISPR advancements have outpaced ethical guidelines and regulations. A governance gap exists, leaving a void in oversight. Addressing this gap is crucial for responsible use. Clear ethical guidelines for CRISPR are paramount. Guidelines should address germline editing, human enhancement, and access. International collaboration is essential for consistent standards. Robust regulatory frameworks ensure safety and efficacy. Transparency and public engagement build trust. Balancing innovation and risk is essential. Decisions made today shape the future of genetic manipulation. Responsible use of CRISPR can enhance human well-being. Preventing misuse is crucial to avoid exacerbating social inequalities. Section 1. A future free of genetic disease. The promise of genetic cures. Imagine a future without inherited diseases. CRISPR offers the hope of rewriting faulty genetic code. Diseases like cystic fibrosis and Huntington's could become relics. Genetic cures are now a tangible goal. Clinical trials using CRISPR are underway for various disorders. These trials represent steps toward overcoming genetic diseases. CRISPR could also prevent diseases altogether. Pre-implantation genetic diagnosis screens embryos for disorders. Combined with CRISPR, it could break the cycle of inherited disease. Genetic cures offer longer, healthier lives. They alleviate suffering and economic burdens. Families could see a brighter future free from genetic disease. Enhancing human potential. Pushing the boundaries of biology. CRISPR's potential extends far beyond fixing what's broken. It offers the possibility of enhancing human capabilities. Imagine enhancing cognitive abilities, boosting immune systems, or extending lifespans. CRISPR could enhance muscle mass and recovery. This impacts athletes, soldiers, and physically demanding professions. Imagine Olympic athletes shattering world records, or firefighters battling blazes with superhuman resilience. The potential for cognitive enhancement is profound. CRISPR could enhance memory, learning, and intelligence. This could revolutionize education. Imagine students grasping complex concepts effortlessly, or scientists making groundbreaking discoveries. While it may seem like science fiction, the science is advancing. Enhancing human potential through genetic engineering is becoming a reality. The dawn of personalized medicine tailoring treatments to our genes. Imagine treatments precisely tailored to our genetic makeup. Personalized medicine leverages genomics to tailor treatments to each unique genetic profile. CRISPR plays a pivotal role, optimizing treatment efficacy and minimizing side effects. Pharmacogenomics studies how genes influence drug responses. Analyzing genetic makeup predicts medication response and side effects. This allows precise drug selection and dosage adjustments. CRISPR holds promise for gene therapies tailored to specific mutations. Imagine immune cells targeting cancer cells, leaving healthy tissue unharmed. Personalized medicine shifts healthcare to a proactive, preventative model. CRISPR and the environment engineering a sustainable future. CRISPR offers a powerful tool for a sustainable future. From biofuels to pollution cleanup, CRISPR can revolutionize environmental conservation. Genetically engineered microbes clean pollutants and restore ecosystems. Imagine microscopic cleaners devouring pollutants. CRISPR also helps develop sustainable energy sources. Engineered algae produce biofuels efficiently. CRISPR can conserve biodiversity and control invasive species, ensuring the survival of our planet's rich biodiversity. Section 5. The Genetic Odyssey. Charting the course of human evolution. The ability to manipulate our own genetic code represents a profound moment in human history. With CRISPR, we can control our evolution, shaping our species for generations. This power could enhance our resilience to disease and adapt to new environments. But it raises profound ethical questions. The genetic odyssey is fraught with both promise and peril. Section 1. The Weight of Choice, Our Genetic Legacy. As we stand at the cusp of this genetic frontier, we are burdened with a profound responsibility, the ability to manipulate the very essence of life, to rewrite the code that has shaped us for millennia, is a power we must wield with utmost care and foresight. The decisions we make today about how we use and govern these technologies will reverberate through generations, shaping the future of our species and the planet we inhabit. The allure of a future free from genetic disease is undeniable. The prospect of eradicating debilitating conditions that have plagued humanity for centuries is a beacon of hope, a testament to the power of human ingenuity. Yet, we must tread cautiously, mindful of the potential pitfalls. The pursuit of genetic perfection could lead us down a slippery slope, eroding our acceptance of human diversity and fueling a new era of genetic discrimination. We must remember that the human story is written not only in our genes, but also in the tapestry of our shared experiences, our triumphs, and our struggles. It is this rich tapestry that defines us as a species, and it is this tapestry that we must strive to preserve as we venture into the uncharted territory of genetic manipulation. Section 2. A Shared Responsibility, Navigating the Genetic Frontier Together. The genetic revolution is not a solitary endeavor, confined to the sterile walls of laboratories. It is a collective journey that demands open dialogue, ethical reflection, and the active participation of all members of society. Scientists, ethicists, policymakers, and the public must work together to navigate this complex landscape, ensuring that these powerful technologies are used responsibly and equitably for the benefit of all. We must foster a global conversation about the ethical implications of genetic manipulation, engaging with diverse perspectives and cultural values. What are the acceptable limits of genetic intervention? Who gets to decide how these technologies are used? How do we ensure equitable access to these life-altering advancements? These are not questions with easy answers, but they are questions we must confront head-on with honesty and courage. The development of robust regulatory frameworks is essential to guide the responsible development and application of genetic technologies. These frameworks must be grounded in sound science, ethical principles, and a deep respect for human dignity. They must also be adaptable, evolving alongside the rapid pace of scientific advancements and the emergence of new ethical challenges. Embracing the possibilities and safeguarding humanity. The genetic revolution offers extraordinary possibilities and daunting uncertainties. We can reshape our species and enhance our capabilities, but we must be mindful of unintended consequences. Embrace possibilities with wonder and responsibility. Alleviate suffering and protect our planet. Remember our shared humanity and interconnectedness. Harness knowledge for the betterment of humankind. Engage in thoughtful deliberation and proceed with compassion. The future is unwritten. Let's write it wisely.